Hello, I'm Nathan McDonald. I'm going to be moderating the second session on blood-brain barrier opening. My co-moderator is Richard Price, and we have 7 speakers here today to answer questions about their talk. So I'm going to jump right into it. Start with a question for Katie, who gave a talk about transplanting mitochondria after stroke through BBB opening. We had a question from the chat was, how soon after the stroke did you treat the mice? And is that a realistic time frame, whatever you used?
Great question. So we treated the mice with FUS about 2 to 3 hours post-stroke. So they received the middle cerebral artery occlusion for about 90 minutes. And then we're allowed to recover for a couple hours and then moved over to where we were completing the FUS treatments, which we believe is a pretty reasonable time point. You'll see a lot of stroke studies that actually treat in advance of injury, which isn't unfortunately particularly applicable right now. But within a few hours, a lot of patients are realizing something is wrong and on their way to the hospital. But we'd like to look at a longer time point treatment as well, maybe 24 or 48 hours after injury.
Have you thought about trying this after TPA?
We have thought about it. We haven't tried it just yet. But yeah, that would be a really interesting experiment to do because the cerebral artery occlusion is removed and there's reperfusion following the removal of the filament. We don't have the blockage that we need to open up with the TPA, but we could certainly look at other models as well.
Okay, thank you.
Thanks.
A question here for Yutong. So has the 50 nanometer nanoparticle been validated? So this is essentially the optimal size in your model. And do you think that the same size dependency could be translated to other types of nanoparticles, for example, a gold nanoparticle?
Thank you for the question. For the current study, we just used our mathematical model to quantify the system of the brain microenvironment and for the nanoparticle delivery, and we use our mathematical modeling to generate a hypothesis that optimized for the nanoparticles. So yet we haven't experimentally tested this theory, but we'll be very interested to do the experiment and test if the theory works. And for the question about the gold nanoparticles, in theory, for the physical side, 50 nanometer would be it would be the best for delivery under focused ultrasound into brain and brain tumors. But again, we haven't tested yet.
Thank you. So let me ask you a follow-up too. I was really interested in as a former nanoparticle transport enthusiast, I really enjoyed your talk. So I was really interested in how you implemented charge into your model. So you show that different charged nanoparticles can behave much differently. How is that actually implemented in the model? Do you have a homogeneous charge in the system, or is it distributed in some other manner?
So that's a great question. So in terms of the computational cost, it's not realistic for us to incorporate the electrostatic field in the model, but to account this effect, the charge will we take into account the effect by the electrostatic hindrance through the porous medium, which we use to model the vessel wall and the interstitial space of the tumor.
Thank you.
All right, I have a question for Robin. The question is about with your cavitation controller. It seems that the cavitation dose would be very dependent on the local population of microbubbles. How would you control with your, the dose that you're using changes in their number or density within the focus?
Yeah, so we didn't directly look at varying the concentration dose of the microbubbles in this study. But what I would expect is that if we were to inject a higher dose or a lower dose using this controller system, what would change is basically the sonication time. So if we were to increase the microbubble dose, then what we expect is greater cavitation signals from each sonication pulse, making us reach the desired doses in a faster manner. And then also in the other direction, if we're in a lower dose or infusing the microbubbles, we would actually have lower cavitation signals per pulse, and then actually have a longer sonication duration. So if we were to vary the concentration, we would also then, as a result, change the sonication duration would be changed.
How will you be able to tell if, at the focus, you're in an area with a low vascular density, that a low dose that you're picking up is from that or from being at a too low of a pressure amplitude?
Right. So that's one of the kind of downfalls with just PCD. It's a 1D signal. We can't exactly identify where we're getting the signal. That's why something we should start looking toward using the controller systems with is also for PCI, or passive cavitation imaging, giving us 2D images, allowing us to further specify where we want to look at the cavitation so that we can further identify exactly where the signal is coming from and further improve our prediction of the bioeffects following BBB opening.
Thank you. Richard, did you want to question Alexander, or do you want me to?
Sure. He's used to it. So Alex, there's actually two questions for you here. I'll ask you the first one first and you can answer, and I'll try to unravel the second one. So the question is about differential gene expression in the microglia between 0.2 and 0.4 megapascals. Are they consistent with the pro-inflammatory phenotype in the microglia?
Yeah, this is a great question. We have looked into the nature of the genes, and while many are related to inflammation, such as CD68 and Ly86, there's not a clear pro or anti-inflammatory phenotype that we can assign to them. Interestingly, the transcripts we did detect overlap significantly with gene profiles associated with the resolution of inflammation after TBI in mice. And if you consider the time point that we're looking at, it's 48 hours after FUS. So this is pretty reasonable to expect those activated microglia to start to return to their resting state.
Nathan, do you want to ask him the second question? Maybe we should move along to the second talk of Alex's, just to make sure everyone gets some time here. Yeah.
Sure. So there was one question. Did you see any changes with microbubbles and FUS that and how it might relate to the clinical trials? So you were looking at anesthesia effects on gene transmission. So when we're doing patients, they have they're awake, but they also may be taking pain medications. Do you think that could have any impact on the outcome of the procedure?
Yeah, this is a really good question. It's one we talked about quite a bit. Unfortunately, we don't have the capability in-house to reliably open the barrier in mice without anesthesia. But I think it's a super important question. And one thing that we wanted to highlight is that the anesthetics really variably can affect things like the vasculature, the kind of baseline inflammatory response. And so this is true for pain medications too, and a lot of the other different drugs patients might be on. So it might be worth considering just in the context of clinical trials. It's a great question.
Thank you.
Okay, so the next question is for Ying, and I actually had the same question. So is the antibody, the trastuzumab, going to be injected before or after barrier opening, and why have you made that decision?
Thanks for the question. So we inject our drug immediately before BBB opening. So right before the patients are prepared and entering the device, we give them the infusion of the drug. Our thinking with that protocol is that we want the highest amount of drug to be in place while the sonications are happening. The thinking is that there's a fast phase of leakage with the initial sonication that might get more of the drug into the parenchyma.
Do you plan to correlate this with MR or cavitation dose, the drug delivery?
Yeah, so one of the interesting aspects of this study is that we are radiolabeling the drug with indium-111. So the spatial resolution of SPECT imaging isn't very high, but we'll try our best to kind of localize the amount of cavitation dose received with each lesion and how much more of the drug we're getting through to that particular lesion.
Thank you.
All right, I have a question for Sophie. Have you looked into the acoustic emissions during the short pulses versus the longer bursts? And if you see any differences in terms of their energy distribution? And also, do you think that the RASP approach with the short pulses will allow for delivery of things with large molecules such as proteins or viruses or nanoparticles?
Yeah, so in terms of the emissions, we have looked at this. We've got both passive cavitation detection measurements, and now we're doing passive acoustic mapping as well. In terms of the energy, we see a lot higher energy with the longer pulses, and also more signs of towards the inertial regime of cavitation with the long pulses. Whilst with the rasp, the short pulses, we don't get that at all. In terms of the size of the molecules, this is something that we're investigating at the moment. We're actually looking into delivering 100 nanometer liposomes at the moment, and hope to publish on this soon. But yes, they can be delivered with RASP. It's a question of optimizing the sequence slightly so that we can get larger molecules across.
Thank you.
Okay, so next question is for Mehmet, and it's actually, again, the same question that I had. You performed your studies at 2.5 megahertz. Have you repeated them with lower frequencies that are closer to clinical usage? And if not, do you expect there to be any differences?
Unfortunately, no. We only tried with 2.5 megahertz, but I would expect similar result even with low frequency. Because after one time opening the BBB, we would see several times of this seizure-like activity happening in the same animal.
All right, I have a question for Jersey. So it was a very interesting talk about designing viral vectors that cross better the blood-brain barrier with ultrasound. And what are the properties of the viral caspid that you think are essential for designing them to cross the BBB after ultrasound and transfecting neurons?
Yeah, thank you for the question. I was reading that and trying to think of this myself. So the short answer is we don't know, because it's a random selection. So we make random mutations, about 1.3 billion of them, and then select a small subset based on what we observe. So I'm not even sure we're improving the crossing after BBB opening, or is it that we're, for example, improving the transduction in the brain without affecting the crossing? I would expect that because these viruses are all the same size, they would have very similar crossing capability, but that's not the case. So I'm actually beginning to wonder if there's some biological mechanism that affects that, but we haven't done any mechanical studies, any mechanism studies. It's all based on the selection. So
do you think there might be any issues? Some of the viral vectors have been reported to be, species or even strain specific. Do you think there might be any? Have you tried other strains of mice with your vector?
Yeah. There is a plan for it. So we started doing that. I think Caltech team is doing that right now. And they're trying a non-human primate. So we'll see how is that going to go. I actually have a plan of writing a whole grant on it, trying to compare which different species are affected by the passage and which are not. And hopefully also optimizing, finding a virus that can work in every species. But that's a problem. And because we've only have the example of PHPB and PHPB, which are those viruses that cross the BBB on their own without focus ultrasound, then we don't really know whether that was a fluke, that it only works in one mouse and not the other, or is it something that's going to be the case for other viruses. Usually AAVs work with different efficiency in different species, but they retain some of their activity.
Thank you. So I think now we're going to go into the bonus round where we're asked questions.
I actually have a follow-up for Jersey too. It's a related question. So in your study, you used a synapsin promoter to drive pre-expression in neurons specifically. Do you think that if you were interested in trying to target a different cell, like an astrocyte, and you were using a cell-specific promoter in another kind of cell, that some of the same candidates would pop up that you got in your screen, or do you think it would be totally different? What's your gut feeling on that?
That's a cool question, because that could tell us whether it's more of a transfection related, right? Or whether it's more of a passage related advance, right? So I like this idea. That's interesting. You would probably, so in reality, what would happen is you would get a bunch of viruses, and because you are really severely subsampling and you are not really getting all the candidates during the screen, you get a small section of them, then you would probably arrive at different serotypes. But I think that the viruses that we have done, we tested their selectivity for cells, and they seem to be selective for neurons, which makes me think that we would probably find other viruses if we were to target, let's say, glial cells, because we actually have more of the neuronal selectivity than we would expect just based on, let's say, AV9. I'm forgetting the number top of my head. We just got this data a couple of weeks ago.
Mm-hmm. I think Alex's first single cell abstract is going to be interesting to look at with respect to your work here.
Yeah, I think so. I watched the presentation. I
have a question for Sophie. A group in Sunnybrook recently just published a paper comparing very similar schemes, and they seem to have come to sort of the opposite conclusions than what you're seeing. I was curious to see what your comments are about what might be different between these studies that are looking at the very short versus longer bursts.
Yes, of course. I think the main difference here is the microbubble dose that we're using. We haven't tested RASP at a clinical dose yet, but my feeling is that RASP only works if you've got a higher dose of microbubbles being injected. And what Sunnybrook has done is they've repeated almost everything exactly the same, apart from, I think the frequency is 1.5 megahertz instead of 1 megahertz, and the bubble dose. They've used a clinical dose, I believe, and so it is possible that if you're using that amount of bubbles, you're not going to get the same effects, the same benefits that RASP can give.
Okay.
I have a little more detailed question for Robin, actually. So Robin, I didn't see gfap or IDA1 in your abstract or your talk. Were those, and the reason I asked specifically about those proteins is because they're common markers for astrogliosis and microgliosis, respectively. Were they on the array? And if they were, I assume that they were not significantly changed in your analysis. Is that correct?
Yeah, so for this array, it wasn't part of the panel, yeah, so we didn't look at it, but we definitely have work going on right now, specifically staining for those and looking at the inflammatory response following BBB opening. But yeah, unfortunately, the array we used didn't have those two markers.
Okay, thank you.
I have a question for Yin Meng about the Herceptin study. In our institution, when I've discussed this with the neuro-oncologists, they're not as enthusiastic about this idea for a couple of reasons. One, that patients are being aggressively treated with gamma knife when they have recurrent mets. And there's also many ongoing trials with breast cancer. Have you run into any issues in recruiting patients for your trial? And do you think there could be a role of combining the Herceptin delivery with radiation?
Yeah, absolutely. That's a great question. So the recruitment for this trial has been very, very slow. I think this trial has been open 5 years ago. So more recently, we had more interest. It kind of depends on the mindset of the oncologists who are collaborating with you as well. Certainly, we find that the patients who are more interested in the study are ones who have maxed out a lot of the other options like radiosurgery, radiation therapy, and surgery. So these patients I presented, they have one of them had a brainstem adjacent lesion, and that lesion had been radiated many times and had surgery twice. So she had basically maxed out all her other options, and she was actually the first patient recruited into the study. So it's a difficult trial to do, and I think a close working relationship with your radiation oncologist is probably the best way to do the study.
All right, thank you.
I have a quick detailed question for you, Tong, as well. If I understand correctly, you simulated an injection difference, a timing delay between barrier opening and nanoparticle delivery about 8 hours. Is there a reason why you chose that delay, which is a lot longer than I think a lot of people are typically using?
So the 8 hours is the time. So we inject the nanoparticle actually right after the blood-brain barrier disruption, and then we sacrifice the animal to analysis the distribution after 8 hours. And this is based on our in vitro study about the time point for the cancer cell uptake.
Okay, I misunderstood that then. Thank you.
All right, we have 30 seconds left. I have one question for Alexander on anesthesia. This is maybe a naive question, but these anesthesia agents already cross the blood-brain barrier. But do you think that we could be enhancing the local delivery of these anesthesia agents, and maybe that could explain some of your results?
Yeah, that's a really good question. And agree. There's definitely some evidence for that, but it's still actually an active area of research as to which anesthetics cross most effectively and what their distribution is like. So I think it is certainly possible that blood-brain barrier is even further enhancing that. Anecdotally, we do sometimes notice that the FUS-treated mice will take a little longer to wake up. But interesting question.
All right, thank you. And I think we are out of time. I want to thank all of the presenters today. And if you have any more questions, please contact them directly or use the chat feature on the sessions. Thank you.